Yeah, thank you very much for inviting me to talk here. I'll jump right in. Okay, so this conference, we've already heard a bit about this idea of transhydrogenated FIP, the idea that you can use these ruthenium CP star catalysts to transhydrogenate as opposed to the more conventional cishydrogenation. And we've been using this idea, which I think was actually shown in the context of FIP back in 2001 by Joachim Bargon. But we've been kind of optimizing these reactions and using them to produce this molecule hyperpolarized fumarate. And I've been doing that with these people and of course now many others. Let me see if I can move that. So I can say a little bit about molecule fumarate and why it works so well in the context of FIP and for pole sequence development. So the protons are in the near equivalence regime. And that means that the proton-proton J coupling is larger than anything that breaks the symmetry. What breaks the symmetry in this case is the difference in couplings to the carbon-13. But because this coupling is larger, it means the protons, to a good approximation, remain in the singlet state. Well, what this means is that we don't have proton T1 relaxation during the chemical reaction, but instead we have singlet relaxation. And that's a few tens of seconds. So it means you can do quite long chemical reactions and you can even transport this molecule around the laboratory while it's hyperpolarized, and you're not losing too much polarization while doing that. And then finally, there's no other spin active nuclei present in the molecule. It's really just the three nuclear spins, the 2 protons and the carbon-13. So we have highly efficient polarization transfer because it's not being diverted to other spins in the molecule. And then why is this molecule of interest at all? Well, it's used in, they polarize it with dissolution DMP and use it for hyperpolarized imaging experiments. So you can observe the conversion of fumarate to malate in the body, and this is a marker for cell necrosis. So this is kind of exciting. It's used in dissolution DMP for this purpose. I'm hoping that it might be that more uses of this particular biomarker are discovered down the line, especially if we can polarize it by FIP, and that makes it much more amenable to work with as a hyperpolarized biomarker. Okay, so here's what the experimental setup looks like that I'll be for the experiments that I'll be describing today. We have a reactor here. This is like a steel reactor where we can bubble hydrogen in. So we perform this hydrogenation, and we're going to use a magnetic field cycle to transform the proton single order into carbon magnetization. So here you can see that whole setup. So a chemical reaction happens up here, and we can eject the sample down into a magnetic shield to perform the field cycling. Here are a couple of pictures of what that actually looks like. So you see this insulation wrapped around the reactor, so it doesn't require too much power to maintain the high temperatures. We work at about 85 or 90 degrees Celsius. And you see here a little microfluidic valve. So what this means is we can just, we perform the whole reaction under pressure, and then we just open this valve and the solution is ejected rapidly into the magnetic shield. Okay, so here are the experimental conditions we'll be using for the experiments that I show in this presentation. I'll just highlight this one actually. This is the flow rate is quite high. Well, that turns out to be really useful because if you can mix up the solution very efficiently and increase the surface area that's in contact with the gas, the hydrogen gets into solution much faster. Okay, and then here is a little sequence. So we have our reaction, which is performed in Earth's field, and then we eject the sample from the reactor to perform the field sweep. We use a constant adiabaticity field sweep profile. So we have like the field ramp is very slow at the position of the level anti-crossing, and it can be more rapid when you're away from the level anti-crossing. And then we carry the sample across the lab and inject it into a benchtop NMR spectrometer for carbon-13 signal detection. So we can repeat the experiment a few times and we vary the bubbling duration. And what we see is that the concentration shown in green, this, of course, that increases as you increase the bubbling duration. Well, that would level off after a while, but we don't see that up to about 90 seconds. And the carbon polarization, which is shown in blue, decreases, although not too much. And again, that's because the proton singlet state is quite long-lived. So it means you can have reaction durations of some 60 or 90 seconds. You're not really losing too much polarization that you get out at the end. So by multiplying these pairs of points, the concentration of fumarate and the polarization, we extract the molar polarization, which can be thought of as the concentration of unity polarized fumarate molecules. And we see that that increases over time and levels off as you have this competition between the reaction rate and relaxation. Okay, so we can produce solutions of relatively highly polarized and highly concentrated fumarate, but of course they're contaminated with catalyst and unreacted precursor and reaction side product molecules. So we introduce this precipitation step. So the second side of the experiment, we have this Halbach magnet and a sintered funnel down here, so that we can extract the solutions of hyperpolarized fumarate. We inject them over here and perform a precipitation, and then we can extract the pure fumarate solutions for applications. So I can show you what that looks like. What we have is HCl sitting on a glass sinter, and we use HCl because, well, it turns out fumarate precipitates if it's in acidic solution. So we can inject the fumarate into the HCl, precipitates as a solid, and then we can filter off the reaction solution. We can wash the crystals, and then they're just sitting on the sinter as a pure solid, and they can be redissolved in, well, we use NaOH because that redissolves the crystals very rapidly. But in principle, you would use a buffered solution if this was for biological applications. So you redissolve the fumarate crystals in solution, and then you can extract them. You extract the hyperpolarized solution. And so not only does this technique purify the fumarate, but you're redissolving the fumarate at a chosen concentration. And I think that's, well, that can in principle be very useful. I have to thank George for helping come up with this procedure, and Peter for helping design and make the small Halbach magnet that we're using. Okay, so there's our experimental setup. And then what we're going to be doing is we perform the fumarate reaction. We'll take one control sample, and with that we can measure the polarization level and the concentration. And we'll take the rest of the sample to be purified. And now here's an important caveat. For this work, initially we've been using a solution of unpolarized fumarate. So we have highly concentrated, non-polarized fumarate waiting in this syringe, such that when we pull in the hyperpolarized fumarate solution, it raises the overall concentration of fumarate. And the reason for doing that is that you have, once you're above about 200 millimolar fumarate, then you have very rapid precipitation. But if you're below that, then it's very slow. So if we run this experiment, we run the same experiment 3 times over, and we see that there's very little difference in the carbon polarization of the control sample and the purified sample. So this is good news. It looks like the precipitation procedure doesn't cause any inherent loss of polarization. If we do the same thing, but we don't have that Halbach magnet, well, then we just have no, then we see no carbon polarization of the fumarate that went through the precipitation procedure. Okay, so overall we have, when we do this procedure, we end up with 25% carbon polarization on 63 millimolar fumarate in solution, with the caveat being that it's actually 350 millimolar in total because we'd added this unpolarized fumarate. And here we have the T1 times of the 2 solutions in the high field magnet. Oh yeah, we think the difference in the T1 might be because of the difference in oxygen content. Essentially, the control sample was very effectively degassed by the reaction, but the purified sample is redissolved in just a deuterated sodium hydroxide solution that wasn't so effectively degassed. And then this is this is the result. So we have our control sample, purified sample, and in carbon-13 spectra we see some extra peaks in the control sample, of course, from unreacted precursor molecule and presumably some reaction side products here. And we don't see those in the purified solution. And with ICP-OES, we've determined the residual ruthenium catalyst concentration to be 16 micromolar, so 0.2% of the initial concentration. And just for a comparison, the sidearm hydrogenation procedure yields something like 30 micromolar rhodium catalyst left over in the aqueous solution. So very, very much comparable with the sidearm hydrogenation procedure. I don't know how toxic the ruthenium catalysts are, so I can't comment on whether this value is low enough for use in humans or not. And so we have a couple of leftover questions. So the first obvious one is, can you precipitate the fumarate directly from solution? It's not really any good if you have to add a load of unpolarized fumarate to make this work, because then, well, you're just diluting the concentration of hyperpolarized molecules. There's also another question about whether the proton singlet state survives the precipitation. We know that the carbon magnetization does, but it's not obvious whether that would also be true for the proton singlet state. And the final question is, can we then store this hyperpolarized fumarate in the solid state? So I can answer that first question immediately. Stefan performed an experiment with where he upped the starting material concentration to 0.5 molar and went to 20 bar of hydrogen pressure and was able to directly precipitate out fumarate from that solution because he formed over 200 millimolar of fumarate with the reaction. So that does seem to work. I'm not doing that in Mainz because it's just, well, it's an awful lot of starting material to be using, but that does seem to work. And then the next question is whether the proton singlet state survives the precipitation procedure. So here I can just show so this is what we've been doing. We use our field sweep to polarize the carbon-13, and then we perform the precipitation at 100 millitesla, and then we go and detect it. And that works. A little tick there, that's great. And then the other experiment would be, forget the field sweep, just perform the reaction, the protons are in a singlet state, and then perform the precipitation, either at 100 millitesla or just in Earth's field, and then do proton detection. Oh dear, I seem to have missed out an important step here. There's a field sweep after the precipitation step so that you can polarize the carbon-13, and then you can do carbon-13 or proton detection. Well, okay, it turns out neither of those experiments worked. We just saw absolutely no polarization whatsoever. So it seems like the proton singlet state does not survive the precipitation step, whether you're at relatively high field or at Earth's field. Probably because it's just not an eigenstate in the solid. I guess the dipole coupling is very large. And then I can just finish up by saying, well, I'll finish up quickly by saying that we are now trying to store the precipitated fumarate in the solid state for relatively long durations. I can just show these results here, where we see that if you decrease the temperature of the solid fumarate, the T1 becomes very long, like thousands of seconds at liquid nitrogen temperature, and probably much longer at liquid helium temperature. So we're interested in pursuing that, in precipitating the fumarate and then storing it in liquid nitrogen or liquid helium. And I think I'll finish up there and say thank you to all of these people who have been helping with this project and related projects over the last years. So thank you very much for your attention.
Thank you so much, Dr. James, for your presentation. I have a question. So when you inject into the syringe, do you use any control field during the sample transfer?
Yeah, so we have a solenoid. I'll show you here you might be able to see so there's a solenoid that provides a field for the sample as it comes out of the magnet. So it sees a magnetic field the whole way. And by the time you're out of the magnet and you're in Earth's field, well, yeah, then it doesn't really matter. Earth's field's high enough that the carbon remains polarized.
Do you know the ideal relaxation field for the apopolarized sample in D2O?
Sorry, could you say that again, please?
Do you know the ideal relaxation field for the apopolarized sample in D2O?
Ideal as in to have the longest possible.
Relaxation field, like the optimum relaxation field for the apopolarized sample.
Yeah, so Bogdan Rodin, Kirill Shebestov, working for Kostya Ivanov, did some studies of the field dependence of the carbon-13 relaxation, and actually saw their results showed it's very insensitive to magnetic field. It just seems that, yeah, as long as you're above a few microtesla, it doesn't really depend on field. And I haven't looked any further into that.
Awesome. Awesome presentation. Do you have, I'm not sure we have further questions here. Yeah. Thank you so much for your time. This is a very nice presentation. Very good work.